Welcome back to Mad Medicine. In this lecture, we're going to be discussing the concept of cellular death. Now, if you haven't already done so, consider subscribing to our channel because your support really means a lot to us and it allows us to keep this content free of charge so that you can prep for your exams for free. So with that being said, let's discuss cellular death by first doing a recap of cell injury. Now, the one thing you need to remember when it comes to cell injury is that our cells have the ability to adapt to a certain level of stress, okay? And they have many different mechanisms that they use to adapt to that stress. Essentially, you can go through hyperplasia, hypertrophy, metaplasia, etc., to be able to handle that stress. But after a certain point, our cells cannot handle stress, okay? They have a threshold. And when you exceed the cell's ability to adapt to this level of stress that they're being put under, when you exceed that threshold, you are going to get cellular injury. So cellular injury occurs when the cell's stress increases is greater than the cell's ability to adapt and handle that stress. Now, there are many different ranges of injury, which we've discussed in previous lectures, but one thing you need to know is that cells get damaged in two main stages, okay? The first stage is the reversible stage. In the reversible stage, the cell actually swells up, and in our last lecture where we talked about cellular injury, we discussed how cells get damaged, and we discussed how cells in the reversible stage are swelling up because the sodium potassium ATP pumps are not working. When you do not have enough ATP, you can't get sodium out of your cell. That's going to cause the cell to swell because water follows sodium. So with that being said, that's the first step. The second step is the irreversible stage. Essentially, this is where you get membrane damage. The cellular, mitochondrial, and lysosomal membranes get damaged, and when that happens, you cannot do anything. The cell will die. Okay? So, how does this actually take place? Essentially, you have a normal cell. The normal cell can go to a state where you have some sort of injury occurring where it is reversible. And because it is reversible, you can go backwards to a normal state. But if you do not remove the stress, the reversible injury will become irreversible. And once it gets to irreversible, no matter what, you will end up with the cell dying. And you have cell death. And there are certain characteristics of cell death that you need to be aware of when it comes to your examinations because you will be tested on these concepts down the road and you need to know them fairly well. So with that being said, let's discuss cell death at a high yield level, okay? So the main thing that you need to know is that the hallmark, the morphologic hallmark of a cell dying is the loss of a nucleus. The nucleus is essentially where everything is going on, right? That's the brain of the cell. And if your brain goes away, you can't function. The cell can't live if the nucleus is dead, if it's gone. And that is what lets us know that a cell has died. But there is a process, there is a mechanism in how the nucleus goes away when your cell is dying. And you need to understand that because it's something you can be tested on. So what is the process? Essentially, the first thing that happens is the process or is the concept of pycnosis, okay? Pycnosis occurs when the nucleus actually shrinks in size. When the nucleus is shrinking, you are going through the very first process. So let's draw it out because people who are visual are going to appreciate this more. The nucleus will shrink in size. Okay, let me make this nucleus a little bit bigger actually. So I'm going to color it red so you can see that the red part is the nucleus and it is visibly smaller. I hope you can tell it's smaller. Now, the next step is going to be karyolysis. In karyolysis, karyorrhexis. In karyorrhexis, the nucleus is going to break up into smaller pieces. Okay, so here's the same cell, but you have the nucleus breaking up, but it's still small, but it's breaking up into smaller pieces. Okay, and then finally, you have karyolysis, where the nucleus is then completely degraded and you have an empty cell. Maybe you'll have like one or two pieces or something, but it's completely gone. You're not seeing any remnants of a nucleus. When you have karyolysis, the cell is completely dead. There's nothing really going on. There's no way to come back. In fact, throughout this whole process, the cell is just dying, but at karyolysis, it's gone. Okay, so when it comes to the mechanism of cell death, you have two main mechanisms you should be aware of. The first is necrosis, and the second one is apoptosis, and we're going to discuss both of them right now. So cellular necrosis occurs when you have death of cells happening at a large scale. I mean, this is kind of a macroscopic or macro scale cell death. Okay, it's not happening at a one single cell level, it's happening overall. How this happens is some sort of exogenous cell injury results in uncontrolled cell degradation. This could be a lot of things. This could be an infection, this could be inflammation, this could be essentially you could have a stab wound that can lead to necrosis. A lot of stuff could happen. You could have decrease in oxygen being delivered to an organ that can cause necrosis, okay? But something exogenous from the cell, something outside of the cell is causing this problem. Now, normal cellular enzymes are responsible for controlled death, okay? Those normal cellular enzymes are inactivated in necrosis, because this is not a controlled cell death. This is happening extracellularly. The cell does not want to die, so those normal cellular enzymes turn themselves off because that's their only way of preventing the cell from being killed off even more, right? So they get deactivated. Now, usually this occurs because of some underlying pathologic condition that's going on. For example, if you have a clot that occludes blood flow to an organ, that organ that is going to be distal to the clot will not get perfusion and it will go through necrosis. It will become dead. It will die. But these cellular controlled cell death enzymes will not be activated. So the key principles when we're talking about necrosis is going to be two main things. Number one, you're going to release intracellular components, obviously because the cell is going to die. The intracellular components are going to be released when the cell membrane bursts. But you're also going to have inflammation. Why do you have inflammation? It goes down to the first point I just said. Necrosis occurs on a large scale. It's not something that's normal. It's not a controlled environment. It's not a physiologic response. It is pathologic. Okay, we have that right here too. We wrote that. It is due to a pathologic process that's going on. And when you have pathologic processes going on, our body can detect that. When we detect those pathologic processes, we try to fight them. One way we fight them is through inflammation. So you will see inflammatory factors. You will see signs of inflammation occurring when you are dealing with necrosis. Now, this is a larger topic that we're going to discuss in upcoming lectures. But the one thing you need to realize is that there are many different types of necrosis, cellular necroses that can happen. It's not just one type. You can get coagulative, liquefactive, caseous, etc., etc. You can get many different types of necrosis. Sometimes they happen together, and certain parts of our body go through certain types of cellular necrosis. We will talk about that in upcoming lectures, but know this, that there are several different types of cell necrosis. So that's the main first concept when it comes to the mechanism of cell death, okay? The second mechanism is apoptosis. Apoptosis is a little bit different. Essentially, it's completely different than necrosis. This is cellular suicide. Essentially, this is going to be controlled. Controlled. There you go. This is also called programmed cell death. Are there two M's? I think there are two M's. Whatever. Programmed cell death. I can't spell. All right. So when it comes to apoptosis is a process that relies on ATP. It actually takes energy to kill a cell. And genetically, you are going to cause the cell to activate different genes to lead to the cell dying. This can be caused by the cell itself or other cells. That's very important because a lot of times we have intrinsic mechanisms inside the cell that our cells can use to detect something going wrong within the cell. And when that happens, our cells can kill themselves off. Now, what happens if you get a extra cellular organism that invades the cell, takes over the cell's functions, and starts reproducing itself? Sounds very similar, right? It's usually a virus. So when some viruses enter the cell, they can take over the cell's machinery, like the proteases and the replication cascade. When that happens, the cell itself does not have the ability to lead to apoptosis. So apoptosis has to be induced by other cells, mainly white blood cells. White blood cells can detect abnormal cell function as well, and they can kill off the cell. Whether or not it's infected doesn't really matter. They have the ability to figure out if a cell is functioning properly as it should, and if it is not, the white blood cell will destroy that cell and tell the cell, you know what, it's time for you to die. You're done. Thank you for your service. Goodbye. So that is very important. Now, this can involve just a single cell or a small group of cells, but it is not happening on large scale like necrosis. This is happening at a very small scale level. So one example of this could be the endometrial lining shedding during menses. During this process, during the menstrual cycle, when menses occurs and their female bleeds, it's happening because of apoptosis. That's why the endometrial lining sheds, okay? Another example of this would be CD8 positive T cell death. T cell mediated death with killer T cells or CD8 positive T cells is one example of extracellular apoptosis being induced when another cell notices one cell not functioning properly. And then finally, embryogenesis. Along the way, when you were being formed into the human being that you are today, when your embryo was being developed, cells were forming, but they were also going through apoptosis. This happens multiple times, and that's how we end up where we are, okay? These are just some examples of cellular apoptosis. Now, we're going to be discussing these concepts in more detail because this is just really an overview of cell necrosis and apoptosis. There's a lot more you need to know for your exams, but you should have a basic understanding to be able to answer majority of the questions. And this is what we're covering right now. So I hope this was helpful. If it was, don't forget to subscribe to our channel. And then if you want to see more free content, go to our website at www.madmedicine.org, where you can find additional educational content completely free of charge to help you with your education and your studying. Thanks.